Thank you, Nishandini. Good afternoon. Good afternoon, one and all. Myself, Dr. S. Manivannan, Assistant Professor, Department of Zoology, Kongrad Arts and Science College, Coimbatore, Tamil Nadu, India. I am extremely privileged to have the opportunity to welcome the 10th day of the International Virtual Conference on Biology in 21st Century, organized by our Department of Zoology. I extend our warm welcome to our respected and most distinguished President, Dr. M. Arichami sir. Welcome, sir. I am very glad to invite our Honorable Dr. C. A. Vasigi Madam, Secretary and Director, Kongrad Arts and Science College, and Syndicate Member of Bharathiar University, and also Convener of Indian Science Congress Association, Coimbatore Chapter. Welcome, Madam. I am also happy to invite our respected Principal and Head, Department of Zoology, Dr. M. Lakshmanasamy sir. Welcome, sir. I kindly welcome to our most respected former Vice Chancellor of Tiruvallur University, Vellore, and the former Registrar in charge of Bharathiar University, Coimbatore, and our Honorable Emeritus Professor, Dr. K. Murugan, sir. Welcome, sir. I would like to welcome Dean Research and Development, Dr. S. Palsamy, sir, and the Dean Academics, Dr. S. R. Madan Sankar, sir. Welcome, sir. I kindly welcome Dr. S. Binukumari, Madam, Assistant Professor and Head in charge, Department of Zoology. I would like to wholeheartedly welcome all the faculty members, researchers, and students from our institution. I am glad to cordially welcome all the participants from other institutions around the globe. On behalf of our department, it's my honor and a pleasure to welcome today's chief guest and resource person, Dr. Ines Miranda Santos. He is a renowned scientist in the field of metabolic engineering and system biology research. Currently, Dr. Ines Miranda is working as a research scientist at the Center for Neuroscience and Cell Biology, University of Coimbra, Portugal, where she is responsible for various cancer research related activities. Ines got graduated from University of Coimbra and carried out her PhD at Molecular System Biology Group in the same university in collaboration with University of the Saarland, Saarbrücken, Germany. She did her MSc thesis at the University of Texas, USA. To his credit, he has published many research articles in the high impact journals, and she also presented his work in various national and international forums. Her area of research interest includes metabolic engineering, system biology, and cancer metabolism. With his rich scientific background, let us invite Dr. Ines Miranda Santos to deliver a lecture on mapping the metabolism of cells. In this current pandemic situation, she is the hot research area around the globe. So, dear participants, make use of this opportunity. Hopefully, this session will be very informative for all. Thank you for the opportunity. I would like to hand over the session to Dr. Ines Miranda. Please, Dr. Ines. Thank you all.
Hello, good afternoon, everyone. Thank you very much for the opportunity and the invitation to present, to give this humble seminar in your meeting. So I'm still finding the way to share the screen, and then with no further ado, I will start my seminar. Okay, so sharing screen, it's here. Okay. Wow, so for some reason, the PowerPoint is not available. Give me, please.
Yes, Ines, take your time, no problem.
When I click to share the screen, the PowerPoint option is not coming. So either I share the entire screen, which is not so interesting, or if I choose to share a window only. Aha, now it's here. Okay, perfect. Perfect. And please do let me know, do you see my screen now? Do you see my first title?
Yes, Ines, it's visible.
Okay, perfect. Okay, so now that these technical obstacles are solved. I mean, we have been meetings online for more than 1 year now, and there is always something or one or another thing that doesn't go as, that doesn't go straightforwardly. Okay. So Dr. Bharti suggested me or invited me to speak a little about the interconnection of, on the one hand, the experimental biology, but on the other hand, the systems biology. So indeed, we were colleagues in Coimbra in Portugal in a group of systems biology where we had to learn from one of the best professors worldwide in systems biology. And this opportunity of this training, at least to me, not only made me learn a lot, of course, but also kind of configure my mindset in the way I look at things. So, title is how to Google Map the cell. Of course, this is a joke because it's a joke. It's an analogy to the way to say that to use that all of us use in our daily lives. Indeed, to begin, I would like to make a short exercise with you. Of course, if this was in a room, would be much more interesting because we could interact more, but nonetheless, let's try. So what if I would ask you guys to draw a nucleus? What would each of you do? Just like that. This is the most of information I'll give you. Please draw a nucleus. So most likely, some of you, sorry, I lost the presentation again. Okay, can you please confirm if you see my slide that says if I would ask you to draw a nucleus, what would you draw?
Ines, actually it is not in the full slideshow. It is just on PPT mode only. It is not in the slideshow mode.
Okay.
Your slide is not moving.
Okay.
I think you are now in the fourth slide. We are not seeing, we are still seeing the first slide.
Okay. I will stop presenting and I will start from beginning.
Yes, please.
Okay, perhaps I should start first the presentation and then I choose what I want to share. Yeah. Okay. So hopefully now you see a full screen slide, is it?
Yes, Ines.
Okay, and let's see if it moves now.
Yes, it's moving. Yes.
Okay.
Go ahead. Thank you.
I am so sorry. Okay, so coming back, so if I would ask you to draw a nucleus, what would you draw? So some of you, perhaps with a more biological background or background in biology, would draw the nucleus of a cell. But perhaps some of you, which are more into chemistry and physics, would draw the nucleus of an atom. And here we are before two completely different things, realities, the nucleus of a cell and the nucleus of an atom, but still they have the same name. Certainly there are historical reasons for this. We are not going into detail on this, but I will come back to this example in no time. Here again, one more example. What is depicted in the diagram? I know it's not, it's very zoomed out, so the font is very small, and this is partly on purpose to highlight the similarities between, on the left hand, the Delhi Metro net diagram, and on the other hand, a diagram of metabolism, of chemical reactions, intracellular chemical reactions. So this is when the word model, or models in plural, come into relevance. So actually we need, so all of this that I've just showed, the drawing of the cell with the nucleus, the drawing of the atom with the nucleus, the drawing of the metro diagram, the drawing of the chemical reactions, intracellular chemical reactions, indeed, these are all models. Models are representations of the reality. Why? Coming back to the very simple, the simplest example, and the most common example of representation and concepts, it's the electric circuit. I don't know if you know, but also the metro diagrams are inspired in the electric circuits drawing. So on the going back to our electric circuit, on the left hand we have a representation of the components of the electric circuit. So without any caption, even a child could say that there's a light bulb, that's a switch, perhaps battery. Most of people are also familiar with the battery concept, maybe not the resistor, but anyway, on the left hand we have drawings, but and we could have this in real life. We do have this in real life, and these are concepts: battery, resistor, light bulb, switch, current, so on and so forth. And on our right hand we have the representation, a standard, almost universal representation of the components of the circuit. So no matter which language or how we are used to draw the components of the circuits, pretty much whoever looks at this representation recognize this as an electronic circuit with that much components. So this was to say how useful models are, but not only for a matter of communication and not only for a matter of communication, but models also are also very useful to systematize the reality, to rationalize, and therefore to highlight patterns that we couldn't notice otherwise, and to recognize new correlations. So this is the model construction, the model designing have been there since almost ever. And a very classical example that we use daily is the Newton's apple. So from one observation, he decides to rationalize it, systematize it, and from there, since then, and from his systematization and rationalization since then till nowadays, we use such rationalization and systematization for purposes and developing other patterns or highlighting other patterns and recognizing new correlations. And now, the most recent is, but already very widespread, is indeed the mapping, the Google mapping. So maps are not new. So drawings of reality, of geography, and of roads and rivers and the orology, like a hill or a valley. Drawings like these have been there since ever. And just a parenthesis, for instance, Portuguese, when they try to find the sea route to India, they also drew many diagrams representing the path and with the details of what the scene, the details of the features of the landscape they were encountering. So, but now with computation, these models and the representation of the reality is reaching to a completely different level, because now with the power of computation, we can integrate much more information and much more parameters than we could do before. And we can also ask the computer to, or we can also ask the algorithm to find us the patterns or the patterns that we couldn't find with the naked eye or at the first sight. For instance, and this is an example, if we want to go from Paris to Brussels, it's around 400 kilometers. When we plan this trip in Google Map, the Google Map suggests us alternative routes according to different criterion that we choose, like the fastest, the cheapest, the shortest. And then it returns a solution for this, a solution for the routes according to our requests, but also according to the parameters that are always being updated from the data collected from the different drivers, such that it suggests, in this case, when I tried this to make this slide, there was 3 possible paths, 3 possible routes. And finally, to come down again to biology, this is also pretty much what happens in the chemical reactions that take place inside the cell. So nature is lazy by nature. So if the nature can do it cheaper or faster, nature wants to do it longer or more expensive. So when we represent the intracellular metabolic reactions in the theme of these seminars, the biology for the 21st century, the taking home message for this part that I would like to pass is that to implement a computational visualization tool of metabolic pathways, meaning of the intracellular chemical reactions, would enhance the integration, the analysis, and the characterization of metabolic patterns according to the most relevant criteria for the cell. So then we set out to, now I will show you a project that I PhD and after, in which we took this knowledge, this concept, and we applied it in the cell. So this is our cell with all the components. It's here just to look good, nothing special, but also to say that the level of complexity of a cell can be have different layers, and so far we are already very familiar with the genome and epigenome level of approach, the transcriptome, the proteome, the metabolome, and the phenome and expososome. This to say that we come from a decade, like 2 decades ago, that the genome, all the attention was placed on the genome as the raw, as the raw potential of the cell. But then we soon realized that the genome doesn't say everything, because not all the genes are transcribed. And then we realized that not all the transcripts are translated into proteins, and proteins and enzymes are not always active. And even if they are active, they depend on the metabolites. And finally, the eventual phenotype is the combination, or this complete depends on this complete pipeline all the way down to the metabolic reactions that actually take place. And so we sought out to model metabolic reactions, the intercellular metabolic reactions. And here we have a drawing, a representation, a model of many metabolic pathways, namely the glycolysis, the TCA cycle, the lipid biosynthesis, the pentose phosphate pathway here, the pyrimidine, gluconeogenesis, pyrimidine and purine biosynthesis. In this representation, we try to include the central metabolism pathways. And then we wanted to apply this to a specific biological question. So we want to apply this to proliferating cells. This is dividing cells. So proliferating cells, we have several examples, from the embryonic cells to stem cells to tumor cells. So all these cells have only one duty, which is to grow and divide, grow and divide. Whereas in embryonic cells, we want cells actually to grow and to divide. In cancer cells or in tumor cells, we rather prefer to interrupt this process. And on the other hand, in stem cells, we want to be able to stop or to drive the proliferation in a way to obtain the proper differentiated tissue. So having said this, to express the importance of the studying proliferating cell metabolism, what we know, this is my question, what do we know? What is the common knowledge of proliferating cell metabolism? So cells, the common knowledge is that they are highly glycolytic. Highly glycolytic meaning that no matter the conditions around them, no matter the oxygen available, no matter the amount of glucose available, highly glycolytic means that cells undergoing cell division, they prefer to take the glycolytic route all the time. So if you recall the drawing of the Google map, where we have several options to go from Paris to Brussels, here the same. So the cell has glucose, and the cell has several options to use this glucose to get ATP, And the cell, so the common knowledge is that the cell always prefers to do glycolysis. And we know glycolysis is not the most efficient, the most yielding pathway in terms of ATP. So what is also happening during cell proliferation, cell division? So these cells are always, they keep undergoing cell division cycle. So they move from G1 phase to GS, G2, M, and then again from M, they immediately start the G1, S, G2, M. And so we hypothesized that metabolism might change over the cell division cycle. So instead of what we are saying, our hypothesis was that instead of having an homogeneous metabolism, the metabolism being the same over the whole cell division cycle, we hypothesized that metabolism may change, meaning that there might be rerouting of metabolic pathways as cells go through the different phases of the cell division cycle. So again, the classical vision, the classical knowledge that's there in textbooks, and it's indeed widely used in proliferating cells approach, and it's even a marker, a biomarker for a tumor, for instance, is that cells are highly glycolytic. The proliferating cells are highly glycolytic, and this is assuming that the metabolism is homogeneous. It doesn't reroute over cell division cycle. And our hypothesis is that indeed the metabolism, if we could measure the metabolism of a specific phase of cell division cycle, we would see that there's a rerouting of metabolism from one phase to the other phase. And what does support this hypothesis that we placed? So there's this observation in yeast cultures. Yeast is it's a microorganism, but indeed it's an eukaryotic cell. So there's this hypothesis that so there's this evidence that when yeast cells are grown in culture, they tend to show sparks of oxygen consumption and then decrease oxygen consumption. So the oxygen density in the broth of yeast culture changes over time. And I'm not going into detail, but I just want to say that detailed studies on this noticed and showed that in each of these phases, so the consumption, the oxygen consumption changes are associated with phases of cell division cycle. So when we can have a population of cells going through the same phase of the cell division cycle at the same time, so when we have a synchronized cell culture, we can observe the oscillations in oxygen consumption, and they correlate with the phase of cell division cycle of that synchronized culture. On the other hand, if we have a non-synchronized culture, which is most of the cases, we don't see any oscillation. But this doesn't mean that at the individual level, at the individual cell level, the oxygen isn't oscillating as well. So approach this, we thought of a methodology called carbon-13 tracing, and we thought of using the synthesis of nucleosides of RNA and the deoxynucleosides of DNA to inspect and to trace the cell metabolism in the phases of cell division cycle. And don't be scared, I will go in more detail in a minute. And of course, our goals were to determine if metabolism is heterogeneous over the cell division cycle, when we don't see when no matter the population is synchronized or not, and to characterize the metabolic flux distribution in the S phase. So for that, we began by building up a model. It's a genome-wide model, so it's based on the genes on the genome of the cell. This model, because it's a 13C approach, it includes the carbon transfer through the metabolic reactions. Then we came to the model design. We had to define boundaries. We had to simplify the network and to establish the variables like the metabolic fluxes that we could measure and those that we had to impose some constraints, like those that are unidirectional, not reversible. And then finally, for the parameterization, we consider the metabolic balance, which is the net uptake of metabolites, and the biomass composition, because if the cell is proliferating, what it does most is to produce biomass to grow. So our model included the glucose input, the glycolysis, the gluconeogenesis, so the glucose input, the glycolysis, the gluconeogenesis, the pentose phosphate pathway here, the TCA cycle here, the glyoxylate cycle, because yeast has glyoxylate cycle, the purine and the pyrimidine biosynthesis, the biomass production, and here you have all the outflows for biomass, the ethanol production, and the storage of carbohydrates. So as we feed the cells with the 13C glucose, the 13C will distribute through as we feed the cell with 13C glucose. So 13C glucose works as an ink. Imagine that you have a bucket of white ink, and you start pouring in it blue ink. Or if you have a bucket of yellow ink and you start pouring on this bucket red ink. So the color of the ink in your bucket will turn into light orange, and as you pour more red ink, the color will become more and more orange until the equilibrium. So this is what happens with 13C labeling. So when we give a substrate that has carbon-13, the cells metabolize the substrate the same way as normal substrate, but we are able to follow the route that the 13C carbon, the carbon-13 takes through the metabolic network. So depending on where the carbon goes, we will be able to find it in particular positions in the biomass. So finally, Because we had a population that was heterogeneous. So we have in the same culture, we have cells in the different stages of cell division cycle. So in our model, we had to consider not only one metabolic map, not only one metabolic diagram, but we had to consider one for the cells in S phase and another one for the cells that are not in S phase. And why is this? Because cells also secrete metabolites to the extracellular medium, and these metabolites that are secreted can be taken up by the cells, by the other cells. So we had to contemplate these two models, these two cell populations, in a way to account for the interchange of secreted metabolites. So with a little bit more of detail now, we chose, as I said before, to look at the carbon-13, where the carbon-13 will end in the nucleosides of DNA and RNA. So this is how it looks like. So this is the glucose molecule with which we fed our cultures, and our glucose molecule has two carbons that are 13C, are labeled with the 13C isotope. And these carbons will go all the way, this glucose will go all the way to through the metabolic pathways, and if depending on the metabolic pathways active in the different phases of the cell division cycle, the 13C will show up in different places in the nucleosides trapped in the DNA and RNA. Okay, don't worry if you are not so following this part so much, because this 13C tracing would be it's actually a 2 weeks course to fully to get the complete knowledge on this, or even more. So far, what is the rationale is, if the metabolism is heterogeneous, so if during the S phase the metabolic flux is different of the other phases, we will see the carbon-13 placed in different places in the nucleosides of RNA and DNA. So this is called labeling. So the labeling in DNA will be different of the labeling in RNA, because DNA it's synthesized only in S phase, and RNA is synthesized through over the cell division cycle. And on the other hand, we took the mitochondrial DNA as negative control, because the mitochondria, the DNA and the RNA, the mitochondrial DNA and RNA, they are synthesized at the same time, so we don't expect to see any difference in the labeling. So just to give a little bit more of detail of this mechanism, so we have here the 13C glucose with these two carbon-13. It will pass into the ribose in the pentose phosphate pathway. It will pass into the intermediates of TC cycle, and these are the precursors of the nucleosides. So this will pass into the precursors of the nucleosides. So we will have a precursor with the nucleosides precursors with the carbon-13, and this labeling will be trapped in the DNA and RNA molecule, such that when we look into the labeling in the macromolecule, we can trace back, as we can see in this example. So in the scenario A, we begin, and in the scenario B, in both, we begin with the same glucose labeled here and here. And if the metabolic flux is different, we will end up with a different labeling here or here. So the carbon-13 will end up in different positions in one case and in another case. So this is just to reinforce what I said. So we expect that heterogeneous if the metabolism is heterogeneous, we expect that the labeling in the RNA is different of the labeling in RNA, whereas if the metabolism is homogeneous, we expect the labeling in RNA to be the same as in DNA. And as a negative control, we look at mitochondria, and we expect mitochondrial RNA and DNA to be the same because they are synthesized all over the cell division cycle. So experimentally, this went like this. We had the cell culture, we extract the nucleic acids, we digest the nucleic acids, we analyze them in GCMS. I'm not going into detail, but this was what allowed us to determine the position of the carbon-13. And then with the position of the carbon-13, we ran this into the computational model, and we could come up with the flux that were active in one case and in the other case. And finally, I'm reaching the end. I think I'm a bit, almost on my time. I'm reaching the end, and here you can see the results of all of this I was saying. So on your left, you have the metabolic fluxes of the S phase. So this, what is in here and in here, this is indeed this same metabolic model. But now we run this model against the, so we use the experimental data with the 13C labeling, and we run the model with that data, and from the place of where the carbon-13 is in the nucleosides, in purines and in pyrimidines, they computed the relative flux through the different metabolic pathways. And so now let's look at in S phase and compare it with non-S phase. So as we can see here, the glycolysis flux is 0.4 in S phase, whereas in out of S phase it's 0.2. The pentose phosphate pathway, the oxidative branch, it's 0.6. This is relative to 1 femtomole per hour per cell of glucose uptake. So the pentose phosphate pathway in S phase it's 0.6. The pentose phosphate pathway in non-S phase is 0.8. The TCA cycle in S phase is 1, and the TCA cycle in, I'm sorry, but you know, it's very hot in Germany. I don't know why, and the touchpad keeps moving despite I don't ask it to move. I'm just moving with the mouse, so that's why sometimes it stucks and thinks I'm passing the slide. So, and the TCA cycle here is 0.2. So as you can see, immediately from here, we can say that the metabolism in S phase is different of the metabolism in the other phases. Surprisingly, there is indeed quite increased activity of TCA cycle during S phase. Not surprisingly, during the other phases of cell division cycle, pentose phosphate pathway is very active, which indeed correlates to biosynthesis of biomass. Just to give you a glimpse of the raw data used to come up to these fluxes. So here is the raw data. This is actually the abundances of the carbon-13 in the different positions of DNA and RNA. I'm not going into detail. And then finally, we ran, we performed an estimation of the actual fluxes of metabolic fluxes in the S phase. Now not a comparative, not comparing to non-S phase. Now it's an absolute quantification. And surprisingly, during S phase, we have some trehalose accumulation, which is storage sugar, and again we have not so much biomass production during S phase, which is very interesting, and we have quite active TCA cycle, or I mean more active than we could expect, considering all the oxidative stress issues. So, and to conclude, this methodology and the approach, it's indeed suitable to profile metabolism, and it could distinguish metabolism of S phase in a wild culture of cells. So we didn't need to do any invasive approaches like synchronizing cells or sorting cells to address the phase-specific metabolism. With the joint use of carbon labeling and the metabolic model allowed us to quantitatively estimate the fluxes in S phase. And so far, the most striking conclusions is that the metabolic fluxes rates change over the cell division cycle, unlike what is considered in the classical approach, where the cells are assumed to be highly glycolytic. And also the metabolism is more oxidative during S phase, unlike one could expect to preserve DNA from oxidative damage. Okay, the last but not the least, of course, this is a joint work of many people, and first of all, my great and strong acknowledgement to Dr. Armin Salvador, my supervisor, Barty's supervisor, and to Elmar Heinzel in Germany, also supported this work and encouraged a lot with all the struggles we had to go through. And then other people in the University of Coimbra, Marta Pinheiro, Silvia Gramacho, Maria João Moreno, and of course the funding that I took during this research project. And well, that's it. I will leave you, and I'm very happy to take questions. I will leave you with the dew of the crown, not this one. And I'm very happy to take questions. I hope it was at least some parts of it were clear.
Thank you very much, ma'am, for sharing your expertise about mapping the metabolism of cells with all of us.
Now the session is open for discussion. Participants are requested to post your queries either in the chat box, or you can unmute yourself to ask your queries.
So Dr. Satyendra Kumar Sinha is asking, I talked about metabolic map in S phase. My query is, what do you think about metabolic map in the case of metabolic cessation? That's a really good question. I'm not sure if I understand what you mean by metabolic cessation, but I imagine that cessation you mean by like a knockdown, or if some metabolic pathway is silenced, or if it's blocked, like when we have construction on a road. So the good thing of the metabolic mapping, these metabolic models, is that you don't have to input such constraints. So if when you run your model with the experimental data, the model will find the routes that were active to produce such those experimental data. So taking this here, taking this slide that I'm showing, if you remember, we began with this representation where all the metabolic reactions have potentially the same flux, so to say. And then when we run our experimental results in this model, we realize that indeed there are metabolic pathways that are shut down. So we didn't need to constrain them as zero. We didn't have to have a priori knowledge on which pathways were active or not. So if a pathway is not active at all, the model will show it. I hope I have answered your question. So the other question is, how are reactive species induced during cellular hypoxia? Well, this goes way out of my expertise. I'm not an expert in reactive oxygen species and in oxidative stress. But during hypoxia, I would say, reactive oxygen species, they need oxygen to be produced. So if there's hypoxia, I don't see like reactive oxygen species to be produced. But if I understand your question, something very different is when after hypoxia you so after hypoxia, the oxygen supply is resumed and is resumed suddenly. So it is known, recently it is recognized that the reactive oxygen species is an equilibrium. So the amount of reactive oxygen species, it's in equilibrium between reactive oxygen species production and their scavenging or their reduction. I mean, Dr. Armindo Salvador, Dr. Armindo Salvador, I was reading another question. Dr. Armindo Salvador is actually a great expert in this reactive oxygen and in oxidative stress. So what I'm saying is what I've learned from him. So the oxidative stress and the abundance of reactive oxygen species is an equilibrium between the production, the scavenging, and the reduction of these oxygen species. And it is known recently, that indeed the protection against oxygen species is inducible, like it is the lactate, it's inducible. So you need to have some abundance of oxygen species to induce the transcription of the scavengings and of the reduction enzymes. So in hypoxia, there's no production of oxygen species, of reactive oxygen species, so the scavenging and the protection is decreased. So then when suddenly you give a bolus or you resume suddenly the oxygen supply, the cells are unprotected, so to say. I don't know if I was clear, but you can ask or Okay, so then I think I'm jumping one question
Thank you.
because this question is coming is in relation with what we are discussing. During hypoxia, you are having less oxygen to accept electrons in the electron transport chain, right? So there is a chance of more free residuals. Well, by free residuals, what do you mean? Like by you don't have acceptors for electrons, so they have to go somewhere? Radicals. Okay, but the radicals, okay, this is all a matter of this is always a chain of reactions. If you don't have the final acceptor, all the reactions upstream will reduce and will eventually stop. You see, so the electrons won't flow through the electron transport chain unless they have one final acceptor. So, I mean, again, I'm saying I'm not an expert in oxidative stress, but this is, I mean, I can give you some references on this, like some people that are more expert, that are actually experts on this, but yeah. So this thing happens with any. This is a matter of how things go inside the cell. So the reactions will take place unless they have a withdrawing reaction downstream. You see, so coming back to repeat what I have just said, if you don't have a final acceptor, upstream the chain of reactions will stop. I'm not sure if I understood, or if I was clear, but please just like type more or speak. Maybe it's easier if we speak. Can we apply, so now this question, can we apply this approach to diagnose or design therapeutics for cancer? So the straight answer is yes, we can. And indeed, we need human resources, we need people interested in this topic to do it. Because this topic, what I have just showed you, is a multidisciplinary topic. We need people from data science, we need people from systems, we need mathematicians because of the optimization, the algorithms of optimization. We need experimentalists, more like me. So this to say that yes, there's lots of work that can be done. This approach is very much in its childhood, and the way I foresee to apply this to diagnose or therapeutics for cancer, actually diagnosis and therapeutics, they go hand on hand with each other. So imagine that we could we can, if we make a model of a human cell, making customized models, it's straightforward, and with this approach we can let's first go to the therapeutics. We can, for instance, extract, when we do a biopsy, we can take those cells, grow them in culture, and test some therapeutics in those cells. And we can see where the therapeutics are acting, and we can test different therapeutics. Or on the other hand, in a more predictive approach, we can take cells from skin, which are skin cells from the patient, so we don't need biopsy. And so these skin cells, they have exactly the same genome of the tumor, and we can test some therapeutics and see which therapeutics would be more effective for that specific genome in stopping, halting the proliferation of those cells. I mean, this is still a lot of science fiction. So ideally I can say where we can go with this, but in practice I foresee lots that this is going to be quite a huge amount of work, and we need people to work in such a field. And again, the problem is always the funding, because these are not fancy things, so it's not easy to get funds for this kind of fundamental science work. And the diagnose, it works the same way. With this approach, we can identify what could be good biomarkers. So this approach may help us to choose biomarkers to be used in diagnose. I hope I have answered your question. Okay, you're very welcome, all of you guys that are acknowledging. I also acknowledge and thank you too for listening to me, and I hope you could learn at least something, and that I got you a little bit more enthusiastic than what you might already be.
Thank you very much, ma'am, for answering.
Hello.
Hello.
Can you able to hear me?
I can hear you.
Hear me? Yeah. Well, presentation. I accept the most of the things, but the metabolism, whether it is the same in twins, if they are S-phase, during the pregnancy, if twins were formed, whether it is any difference between the cell mapping in that embryo.
Well, that's a good question. It's a tricky question. So the mapping, say that again. Okay, so the mapping between the twins would be as different as their genome is. So we know that there are different, so the similarities between twins depends very much on when in the embryology the twins were formed. So twins can be formed at the first division of the egg. Twins can be formed because of the female gamete was indeed a 2 nuclei gamete, and it's fertilized by two spermatozoids. So the difference in the mapping, it all depends on the difference on the genome of these twins. But this that I'm saying is just the basics, because then you have on the genome, as I showed here, where is this? As I showed here, here, on the genome, you then on the genome, you add layers that are specific, of the circumstances of the cell, of the circumstances of the individual. Let me give you an example. Imagine that these twins were absolutely the same with respect to genome. Imagine that their genome was absolutely equal. But post-transcriptional regulation could be different in one or the other. Post-transcriptional, the environment where they are can be different. So this to say that this is one of the strengths of this approach. It begins with the genome and it will end with the environment of the person or the cell.
Thank you, ma'am.
You're welcome.
Thank you very much, ma'am, for answering all the questions asked by our participants. Here, the participants are requested to fill the feedback form which is posted in the chat box.
Once again, I thank everybody on behalf of myself and the organizing members, Department of Zoology, Kungunadu Arts and Science College, Coimbatore, Tamil Nadu, for making this day a great success. Thank you.
Thank you, Ines. Thank you so much.
Thank you, Bharti. Thank you all.
Yeah. The presentation was
Then
very
you
nice
let-
and informative.
And then you let
Informative
me
too.
know. Okay.
Yeah, thank you.
Bye-bye.
And
Thank you. All the
bye-bye,
best.
bye-bye, Ines. Yes, thank you so much.
Bye.
Bye-bye. I have some information to the participants. At 3:30, we are planning to have the valedictory section of the conference. All the participants are requested to join with us as you are doing in all the days of the conference. It will be great if you all join with us for the valedictory function and grace the event. And the feedback link for the today's session will be posted at the end of the valedictory function. So it will be started at 3:30 p.m. That is in the 10 minutes. Please stay with us. Thank you so much.
Good afternoon, Manivanan sir.
Yes, sir.
Sir, good afternoon, sir.
Yes, shall we start?
Sir, waiting for. Hello, sir.
Student sir,
Sir, waiting for
waiting for?
Murugan sir. Murugan sir.
Okay. Volume பாருங்க.
Sir, good evening, sir. Sir, good evening, sir. Murugan, sir.
Nishantini, are you there?
Yes,
Nishantini
ma'am.
Vishnupriya? Yeah, you can
I'm here,
start,
ma'am.
ma. Yeah, you
Okay,
can
ma'am.
start.
Science knows no country because knowledge belongs to humanity and is a torch which illuminates the world. Louis Pasteur. This notion, I agree very good to one and all present here, and I wholeheartedly welcome everybody to the validatory function organized by the Department of Zoology of Kogunada Arts and Science College, Coimbatore, Tamil Nadu.
Prayer is a positive vibe. The good start always begins with the blessings of the Almighty. So I pray the Lord to shower His blessings throughout the entire session. The time has come to ravishingly welcome you all to this program. Now I kindly request our Honorable Assistant Professor and Head in Charge of the Department of Zoology, Dr. S. Binukumari ma'am, to deliver the welcome address. Please, ma'am.
Thank you, ma. Good evening and warm greetings to one and all. On behalf of Zoology Department, I extend a very sincere warm welcome to all of you present here on the valedictory function of International Virtual Conference on Biology in 21st Century organized by Department of Zoology, Kogunada Arts and Science College, Coimbatore, Tamil Nadu, India from 4th August to 13th August 2021. I feel privileged to welcome our esteemed Honorable Secretary and Director, Dr. C.A. Vasigi Madam, distinguished Principal Dr. M. Lakshmanasamy Sir, most valued, respected our Emeritus Professor Dr. Murugan Sir. I also extend my warm welcome to our guest of honor, Dr. Ins Miranda Santos, Research Scientist, Center for Neuroscience and Cell Biology, University of Coimbra, Portugal. And also, I wish to welcome the faculty members of Zoology Department and other institutions, research scholars and UG and PG students of Kogunada Arts and Science College and other academic and research institutions. I am really appreciate and congratulate the organizers of this virtual conference, Dr. Manivannan and Dr. Bharati, for organizing such an informative webinar. I am also congratulate all the participants. Let everyone appreciate the efforts of all the participants who have been participating to showcase their talents. We hope this 10 days international conference help the students to expand their mental horizon to get into self-introspection as well as their achievements. There encourages the students to participate in various events and see their blossom into an individual and help the Indian education system adopt the best of the education system around the world. I hope you all will understand the value of the conference and will enhance it for a better future. Thank you all for being such a patient listeners. Thank you very much.
Thank you, ma'am, for your warm welcoming.
Thank you.
A leader is one who knows the way, goes the way, and shows the way. Now, I kindly request our beloved principal and head Dr. M. Lakshmanasamy sir to deliver the felicitation speech to the gathering. Please.
Thank you, Subhashini and Binukumari.
Thank you, sir.
Good afternoon, one and all present here. Most respected Secretary and Syndicate Member of Bharatiya University, Dr. C.A. Vasigi Madam, Dr. K. Murugan Sir, former Vice Chancellor of Thiruvallur University and former Registrar of Bharatiya University, and Emeritus Professor Department of Zoology, Kungunadu Arts and Science College. Guest organizing committee members, Dr. P. Bharati and Dr. S. Manivannan. Faculty members, research scholars, PG and UG students, participants from Tamil Nadu and other states. On behalf of the management, welcome all for the validity function of international virtual conference on biology in 21st century. On behalf of the management and also of the Department of Zoology, I really appreciate the organizers. Nearly 100 abstract research articles were published. And this is the very initiative for the UG and PG students. Actually, the UG students have a UG project and the MSc students have a MSc project. I think this virtual conference will really be for the UG and PG students. Welcome you all, and I wish the program be a grand success. Once again, I really appreciate the Zoology department for this program. Thank you.
Thank you, sir, for your warm speech.
Now, I kindly request our respected Assistant Professor of Department of Zoology, Dr. Ma'am, to pitch conference. Please.
Good afternoon to one and all with us for the validity function National Virtual on century. Secretary and Secretary Dr. C.A. Vasiki Madam, our beloved Principal Dr. M. Lakshmanaswamy Sir, Honorable Dr. Murugan Sir, and all the faculty members and all the participants of this conference. Myself, Dr. P. Bharati, and my colleague, Dr. S. Manivannan, Assistant Professors and conveners of this conference, and also all the faculty members of the Department of Zoology, Kungunadu Arts and Science College, Coimbatore, are very much pleased to recapping the conference. The conference, entitled International Virtual Conference on Biology in 21st Century, was planned to organize from 4th to 13th August 2021. Inaugural session was started on the 4th August 2021 at 2 with a lecture by Dr. Jose Teixeira, research scientist, CNC, Center for Neuroscience and Cell Biology, Center for Innovative Biomedicine and Biotechnology, University of Coimbra, Portugal. The title of his presentation was Toxicological and Pharmacological Effects of New Molecules. Now, more than 150 participants have joined with us and enriched their knowledge in this session. Followed by that, the second lecture was organized on the 5th of August 2021. The speaker was Dr. Srinath Sridharan, who is a Principal Analyst, Health Services Research, Sengai General Hospital, Singapore. The topic of his presentation was Data Science in Healthcare. All the participants have enjoyed and posted so many questions, and the session was very informative to all the participants, and also it paved the career pathway for the participants. And the third lecture was delivered by Soundararajan Nagasundara Pandian, Assistant Professor, Kungunadu University, Department of Stem Cell and Regenerative Biotechnology, Genome Biology Lab, Seoul, South Korea. And he also presented, his presentation was very informative, and also she highlighted about the DAT fellowship for the students, and also she was talking about the internship opportunity in our university, also the neighboring university which is located in the Germany, and also how to apply and how to approach the professors in the abroad as well as India for internship, for PhD and also postdoc. It was very informative session too. The next presentation was by Dr. Kaustubh Nayak, research scientist, ICGEB, Emory Vaccine Center, International Center for Genetic Engineering and Biotechnology, New Delhi. The title of his presentation was Techniques in Immunology, a Snapshot. He was talking about the techniques which is used for the vaccine development and how the vaccines are developed, especially for the COVID-19. Participants, the session was very interesting, and also participants posted so many questions regarding the vaccine development and also the effects of the vaccine over the COVID-19. And also he was talking about the internship opportunity in the national institutes such as CAFR, NCBI, IASC and ICGEB, etc. It was very informative to all the participants as well. The next lecture was planned to deliver by the Dr. Ramachandra from Bangalore. Unfortunately, we didn't have his lecture due to sudden change of his plan. So maybe probably when we will be getting his schedule, we will organize his lecture separately. Sorry for that. The sixth lecture was presented by Dr. Ines Miranda Santos, who was my lab mate when I was doing my postdoctoral research at Portugal. Research scientist, Center for Neuroscience and Cell Biology, University of Coimbra, Portugal. The title of her presentation was Mapping the Metabolism of Cells, and she was explaining in detail about the both theoretical and the experimental methods of how to map the metabolism in the cells. I hope the session was very interesting and all the participants enjoyed it, and so many questions was posted by the participants, and the session went well and it was very informative as well. And I'm very glad to say this, along with all the lectures delivered by the eminent scientists all over the globe, we also received about 80 abstracts from various national institutes, universities and colleges all over the India, from Kanyakumari to Kashmir. We were very happy to say that all the abstracts were presented by the teachers, scientists, students, everyone. So it was very informative, and also we are very happy to say that this conference gave the platform for the student to present their work and enrich their knowledge as well. And I wish to inform that certificate for the presentation as well as the participation will be sent to the presenters and the participants who have submitted feedback for all the session in a week. At last, I thank all the speakers and the presenter and participants from various countries and different states of India for your active participation to make this event a successful one. Thank you.
Thank you very much, ma'am.
Now I kindly request former Vice Chancellor of Thiruvalluvar University, Vellore, and former Register in charge of Bharathiar University, Coimbatore, and a Honorable Emeritus Professor, Dr. K. Murugan sir, to deliver the valedictory address. Please, sir.
Very good afternoon. Very good afternoon to everybody. Very good evening. First of all, I wish to thank our Honorable Director and Secretary of Kongunadu Arts and Science College, Professor Dr. Vasugi ma'am, and my respected Professor Dr. Lakshmanaswamy, Principal of this college, the head of the department, Professor Dr. Binu madam, the conveners of International Virtual Conference on Biology in 21st Century, being held from 4th to 13th of August, Dr. Bharati, Assistant Professor, Department of Zoology, Dr. Manivannan, Assistant Professor, Department of Zoology, Kongunadu Arts and Science College, all other sister department teaching and non-teaching staff of Kongunadu Arts and Science College, the respected Dean, Dr. Paul Swami, and other Dr. Madan Sankar, Dean of Academics in Kongunadu Arts and Science College, all students and teachers responsible for this conference on biology in 21st century. And I am also very happy to participate, and I am with you for this valedictory function. Few words about this conference, and really this is a very lengthy, very long conference on importance of biology in 21st century. The various experts have given lectures on various topics in toxicological, pharmacological, and neuroscience, and particularly Dr. Jose Texeria from University of Coimbra, Portugal, Dr. Sridharan from Singapore. He has talked about data science in healthcare. Dr. Soundararajan Nagasundara Pandian, he's from South Korea, Konkuk University. He has talked about antimicrobial peptides and related applications, and really participants have received any informations on this particular subject, and really it is just like the refresher course for teachers and also students, and really I am very happy. And this conference, I think, benefited much to the younger generations and young teachers and even the established scientists also. And the conducting conference for 1 day or 2 days, 3 days is also a problem. But the Arts and Science College, Professor Dr. Lakshmana Sami has given very good support to these two assistant professor, Dr. Bharati, Dr. Manivannan, to hold this conference in Kongunadu Arts and Science College. It is a really a breakthrough, and really it is a very, very important, and during our 21st century, a lot of improvements, a lot of advancement has happened at the global level. And one sad thing is we also faced the COVID-19 the problems, and so that the government has taken much step to produce a lot of vaccines, and both national and international agencies have involved to tackle this problem. And our indigenous medicine from India, and particularly southern India, Tamil Nadu, particularly neem and other turmeric and ginger extract and other related herbal medicines have given a lot of support to tackle this problem. And really I am also very happy. And also nowadays the water that is contamination and other thing, and also the microplastics in the ocean and also river and also stagnant water bodies, also very, very giving a lot of problem. The microplastics are also ingested by many water dwelling organisms, and the same animals are also during the food chain, it eats the microplastics and it can move through the food chain. The main question is what happens to the toxins that are associated with plastics, and even mosquito larvae also eats and consume these microplastics, and it can be eaten by other fish and other coffee pots and other thing. It transferred to the various aquatic ecosystem. So that these are all the challenging issues in future. The students can take this kind of problems because the water is a universal solvent, and it is contaminated by various means. That we have to we need very quality and good water for everybody, right from child to old persons. So that at this context, this conference from 4th to 13th has given very good insights to the younger generations. A lot of information. I think I request Dr. Bharati and Manivannan, with support of Dr. Lakshmana Swami, principal of this college. He is a very good zoologist, and they can have a very good book or proceedings, and it can be served. It can be served to the various people involved, and is a very good information. The documentation is a very, very important nowadays, so that Manivannan and Dr. Bharati can have this book on biology in 21st century, and we will also involve to edit, and we can give a very good book to the nation. Congratulations to everybody. Congratulations to the audience also. And also I thank the organizer for giving me this opportunity in the evening, and I wish you all the best. Thank you very much. Good evenings. Thank you.
Thank you very much, sir, for your motivating speech.
Thankfulness is the beginning of gratitude. Gratitude is the completion of thankful.
Nishantini, just a minute.
Okay, ma'am. Okay, ma'am.
Thank you, sir. Thank you for your encouraging words, sir. Thank you so much, sir. Now we are asking the feedback from the participants. The participants can unmute yourself and you can give your feedback about the conference. It will be great if any participants unmute and give your feedback. And some feedback or
Good evening.
Good evening, sir.
To principal, secretary, and all the organizers and the staff members of the Zoology department of the Kongunadu College. I usually attend the webinar, but this webinar is a little bit of difference since it is a well executed and well planned, and most of the sessions are live. I am really enjoyed up-to-date knowledge of the speakers, and the participants also enthusiastically participated here. I make my congratulations for the organizers and the Department of Zoology for conducting a wonderful seminar. Thank you.
Thank you, sir. Sir, may we know where you are from, sir?
I am Associate Professor of Zoology, Department of Botany, St. Joseph's College, Trichy.
Thank you, sir. Thank you so much, sir. Is there any other feedback from the participants? It will be great if you unmute and say to us. It will be encouraging to us as well.
Hello. Am I audible?
Yes, sir. Please, sir.
This is Dr. Srinivas, DIT. So very thankful to organizers. Especially I was inspired by all the lectures, and these are all very useful in different research areas and different areas also. In particular, I was very much impressed with two lectures given by Dr. Zima Kodandraman from Germany and Dr. Kaustu Naik from New Delhi. And these are all very useful in my research work personally. I am very thankful to them. Also thankful to the organizers, and special thanks to the management of KASC. Thank you. Thank you very much.
Thank you, sir. Thank you for your feedback, sir. And also there is a feedback in the chat box from Satvika. Thank you for the opportunity. All the sessions were well organized and all the sessions were informative too. Kudos to the department and Kongunadu College. Thank you, Satvika. And also there is a feedback from the Dr. Bhavvi. That is, all the sessions were well organized. All the sessions were very informative and interesting. Thank you, Gauri ma'am. Is there any other feedback from the participants? Okay, thank you so much for giving you the valuable feedback, and thank you so much that made us to get encouragement to organize such an event in the future as well. Thank you so much. Nishantini, you can continue now. Nishantini?
Okay, ma'am. Okay, ma'am. Thankfulness is the beginning of gratitude. Gratitude is the completion of thankfulness. Now, I request our beloved Assistant Professor of Department of Zoology and convener of this conference, Dr. S. Manivannan, sir, to deliver the vote of thanks. Please, sir.
Thank you, Nishantini. Good evening, all. Myself, Dr. S. Manivannan, and my colleague, Dr. B. Bharati, conveners and Assistant Professor, Department of Zoology, Kongunadu Arts and Science College, Coimbatore. It is a great privilege for us to propose the vote of thanks on this great occasion. The first owe my sincere gratitude to our respected and most distinguished President, Dr. M. Aruchami sir, for his blessing in each and everyday activity. I extend my heartfelt gratitude to our most esteemed Honorable Secretary and Director, Dr. C.A. Vasugi Madam, who encourage and motivate us in all our endeavors. Thank you, Madam. It is a great time for me to thank our most respected Principal and Head, Department of Zoology, Dr. M. Lachanasamy sir, for gracing the conference with your presence, encouragement and support. Thank you, sir. I express my sincere gratitude our most respected former Vice Chancellor of Tiruvallur University, Vellore, and former Registrar in charge of Bharathiar University, Coimbatore, and our Honorable Emeritus Professor, Dr. K. Murugan sir, for gracing the conference with your presence. We are all inspired by your great words, sir. Thank you, sir. I also take this opportunity to honor and thank all the invited speakers from various countries, and we are also extending our gratitude to all the speakers who are shared their expertise and work with us, and for enriching our knowledge. I also thank Dr. S. Paulsamy sir, Dean Research and Development, Dr. Madan Sankar sir, Dean Academics, and Dr. R. Sivaprakash sir, Associate Dean, for their support. I must thank our head in charge, Dr. S. Binukumari ma'am, who is us with all the time and encourages us. I also extend my thanks to all the faculty members and non-teaching members who are the pillars of the successful completion of this virtual conference. I am also thankful to Ms. Nishandini and Vishnu Priya, third BSc Zoology, for their involvement and making the conference as live. My gratitude to all the participants are very enthusiastic. I am thankful to all the participants for attending the conference. We have been fortunate to have some of the eminent persons from academia and industry and utility working in the different area of biological science. I am sure that the participant must have benefited by attending this conference. On behalf of Department of Zoology, Congrad Arts and Science College, and on my own, extend my heartfelt thanks to one and all present here and encouraged to complete the session successfully. Thank you.
Thank you, sir, for expressing your gratitude through your humble words.
Thank you, sir. And thanks to all the participants. And now we can wind up the session. Thank you so much.
Thank you, ma'am.
Thank you, ma'am.
Thank you.
Thank you, ma'am.
Thank you. Thank you so much.